In this video, I want to explain why excessive calorie intake leads to insulin resistance and chronic disease. So, when you eat a meal, your body absorbs the glucose in that meal, which then enters the bloodstream. And when that glucose enters the bloodstream, it enters into the pancreas, and then the pancreas secretes insulin into the bloodstream. And here's the biochemistry of that pathway, but what's important to realize is when glucose enters into the beta cell of the pancreas, it automatically goes through the sequence of events that leads to the beta cell pumping out insulin. So then that insulin gets pumped out into the bloodstream. So when that insulin enters into the bloodstream, it interacts with different tissues, for example, adipose tissue and skeletal muscle, and it tells those tissues to upregulate the GLUT4 transporter, which allows these tissues to take up glucose and use that glucose as a source of energy. And here's the biochemistry. Again, insulin binds to the insulin receptor of the cell. Then it activates this cascade of events, which upregulates the GLUT4 transporter, and now the tissue is allowed to uptake the glucose and use the glucose as a source of energy. However, in type 2 diabetes, these tissues, particularly skeletal muscle, becomes insulin resistant. It's one of the early stages of type 2 diabetes. So when the skeletal muscle is insulin resistant, it's resistant to the effects of insulin. So therefore, again, normally insulin causes the tissue to upregulate the GLUT4 transporter. However, if it's insulin resistant, it is not upregulating the GLUT4 transporter. So therefore, the skeletal muscle cannot absorb glucose. So the glucose is stuck in the bloodstream. So then the glucose enters into the beta cells of the pancreas. And remember, as long as glucose is entering into the beta cell of the pancreas, it automatically produces insulin. So therefore, this will lead to hyperinsulinemia, high amounts of insulin in the blood. Basically, as a way to compensate, you upregulate the amount of insulin you're producing, which then forces the skeletal muscle to finally upregulate the GLUT4 transporter, which can then take up the glucose. However, because the beta cells of the pancreas is increasing the amount of insulin, eventually you reach beta cell exhaustion, where the beta cells stop functioning and they stop producing insulin. So then this leads to low amounts of insulin. And remember, the skeletal muscle was already insulin resistant. It wasn't listening to the insulin. And now you have low amounts of insulin. So then this leads to hyperglycemia, high amounts of glucose in the blood. Because the skeletal muscle is not listening to the little bit of insulin that is in the blood. So therefore, there's very little GLUT4 transporter upregulation. So therefore, you can't take up the glucose, so all the glucose is stuck in the blood. This graph represents the time course of the average patient with type 2 diabetes. But again, the first step is the skeletal muscle becomes insulin resistant. And to compensate, the pancreas increases the amount of insulin that's produced. So we see that here. First, you see an increase in the amount of insulin that's produced. And this insulin forces the skeletal muscle to take up the glucose to maintain a euglycemic state, a normal glucose state. So again, early on, you see an increase in insulin to make up for the insulin resistance to maintain a normal glucose state. However, eventually, you have beta cell exhaustion, where the pancreas stops producing insulin, so then you have a drop in the levels of insulin. So as you have a drop in the levels of insulin, then you start to see an increase in blood glucose. And something important to realize is when you're seeing an increase in your blood glucose, that means you're already pretty late in the game. You're already pretty late in the time course of type 2 diabetes. But something also important to realize is this entire process began when the skeletal muscle became insulin resistant. So why does this happen? What causes the skeletal muscle to become resistant to the effects of insulin? What is the biochemistry that leads to this? When you eat calories, our body wants to store the energy in those calories in our adipocytes as triacylglycerides. However, if you keep on eating excessive calories, your body wants to store all those calories in our adipocytes. So the body adapts. We go through adipocyte hypertrophy, where the adipose cells grow in order to accommodate and store all that energy. However, if you keep on eating, you overwhelm the adipose tissue's ability to hold on to these triacylglycerides and this energy. So then your body starts to go through this uncontrolled lipolysis, where essentially these triacylglycerides get ripped apart to release free fatty acids that enter into the bloodstream. So again, these triacylglycerides, they get ripped apart to release these free fatty acids which enter into the bloodstream. Also, adipocyte hypertrophy is very inflammatory, which releases inflammatory cytokines into the bloodstream like TNF alpha and IL-6. And something important to realize is we all have a different genetically encoded fat threshold. For example, some people, if they just barely overeat, they overwhelm their adipose tissue's ability to hold on to those calories and to store those calories, and they start to release that energy by going through uncontrollable lipolysis, hydrolyzing those triacylglycerides and releasing free fatty acids into the bloodstream. While other people, due to their genetics, their adipose tissue can very safely store and hold on to all that energy. It doesn't matter how much they eat, they can store all those calories in triacylglycerides in their adipocytes, and they have a very high fat threshold, where it takes a lot of calories to overwhelm their adipocytes until they start to overwhelm the adipocytes so much that they can't store any more calories, and they start to release some of those calories as free fatty acids into the bloodstream. However, what happens once we release those free fatty acids into the bloodstream? So when we eat calories, our body wants to save and store that energy in those calories. So we store that energy in our adipocytes. However, if you keep on eating excessive calories, you overwhelm the adipocytes' ability to safely store that energy. So once you keep eating calories and you overwhelm the adipocytes, they start to release free fatty acids into the bloodstream. But again, that's precious energy that we want to save and store, so those free fatty acids and that energy starts to store in other tissues like skeletal muscle. So we store that energy in skeletal muscle in the form of intra-myocellular lipids. However, this is a problem, because essentially what happens is the skeletal muscle starts to think, hey, we have energy. We have a form of energy in these intramyocellular lipids, so therefore we do not need glucose. We don't need glucose. We don't need to listen to insulin to upregulate the GLUT4 transporter to take up glucose. We don't need to do that. We don't need the GLUT4 glucose transporter because we don't need glucose. We have a form of energy in the form of these intramyocellular lipids. So let's save that glucose for the brain. And if you're curious, here's the biochemistry. So again, the adipocytes, they get overwhelmed and start to release those free fatty acids into the skeletal muscle. And when those free fatty acids enter the skeletal muscle, they form these fatty derivatives like ceramides and diacylglycerols, which then we can see the biochemistry of essentially how they inhibit this GLUT4 expression pathway. They lead to insulin resistance. And again, this makes sense. The skeletal muscle has a source of energy as these lipid source of energy, so therefore we don't need the GLUT4 transporter. We don't need to listen to insulin. It becomes insulin resistant. We downregulate the GLUT4 transporter because we don't need glucose. We have a form of energy. And also, it's important to realize that those inflammatory cytokines can, through different mechanisms, also contribute to insulin resistance. And this also explains why patients with lipodystrophy have terrible insulin resistance and diabetes. So basically, these patients with lipodystrophy, they have a very low fat threshold. They essentially don't have adipocytes. So when they eat calories, they cannot safely store the energy in those calories in those adipocytes because they don't have adipocytes. Or they have very little adipocytes because they have a low fat threshold. So when they eat calories, the energy in those calories are stored in other tissues, like in the skeletal muscle. So the skeletal muscle thinks, hey, we have energy in the form of lipids. We don't need glucose. So they have terrible insulin resistance. And a way you can think about it is they have a low fat threshold. We can also start to store this fat in our liver, and we call this NAFLD, non-alcoholic fatty liver disease. And it's important to note that this is a very complex disease, and it's possible that branched-chain amino acids play a role, it's possible that fructose play a role, along with many other factors. However, this uncontrolled lipolysis also leads to free fatty acids being stored in our liver. And we know the liver gets rid of these triglycerides in the form of these VLDL particles. These VLDL particles enter into the bloodstream to transport triglycerides and to release free fatty acids to enter into the peripheral tissues. And as they do this, they get degraded into IDL particles and then eventually LDL particles. And then eventually these LDL particles get taken up back into the liver by interacting with the LDL receptor, and then they enter back into the liver. And it's important to note that these lipoprotein particles, they have this ApoB protein, and it's this ApoB protein that interacts with the LDL receptor to allow them to be taken back up into the liver. A very important concept is residence time. For example, if the liver secretes these triglycerides in the form of VLDL, and then they very quickly release those triglycerides as free fatty acids to enter into other tissues to form IDL and then eventually LDL, and then very quickly get taken back up into the liver, that would be a very short residence time. However, in type 2 diabetes, the high insulin in the lipolysis leads to a high residence time. Essentially, these factors affect lipoprotein lipase and ApoC3, where essentially the liver secretes these VLDL particles, but it's very hard for the VLDL particles to release their free fatty acids, so they stay in the blood for a long time until they form into IDLs, and then LDLs, and then they stay in the blood for a long time until they're finally taken up into the liver. So that would be a high residence time. And it's important to note that a high residence time is associated with type 2 diabetes. For example, if these LDL particles stay in the blood for a long time, it's more likely that their ApoB protein will become oxidized. This protein gets oxidized, and that's just natural, unregulated chemistry. That's just inevitable that proteins, when they stay in the blood for a long enough period of time, are going to go through unregulated chemical reactions to get oxidized. And it's important to note that it's this oxidized LDL that gets taken up by the macrophages to form plaque in coronary artery disease. One way to think about it is as this ApoB gets oxidized, it turns into a foreign molecule. It's not normal ApoB protein, it's a foreign oxidized, chemically changed ApoB protein that then the macrophages sense as foreign, and they take it up, and that forms the foam cell, which then eventually leads to plaque. But it's very important to emphasize that there are many factors that contribute to coronary artery disease. A very important factor is the amount of LDL receptors that are on the liver. For example, if we have very few LDL receptors, then these LDL ApoB containing particles are going to stay in the blood for a very long time until they're finally able to bump into an LDL receptor to be taken up into the liver. So if you have very little LDL receptors on your liver, then you're essentially going to have a higher LDL. These LDL particles are going to be stuck in the blood until they're finally able to bump and get taken up. So this can be genetic. For example, familial hypercholesterolemia can result in very little LDL receptors on the liver. And we know these homozygous familial hypercholesterolemia patients, they get coronary artery disease and myocardial infarctions very early, maybe even in their 20s. So that tells you that you need to be in this high ApoB state for up to even a few decades until coronary artery disease progresses, But we know that familial hypercholesterolemia, these genetic conditions, can increase coronary artery disease. We also know that eating saturated fats enter into the liver and they get metabolized into cholesterol. And when the liver has these saturated fats and cholesterol, they think, hey, we have plenty of cholesterol, so we don't need the cholesterol in these LDL particles in the blood. So this sends a signal to downregulate the LDL receptor, so you have very few LDL receptors, so that also increases this ApoB particle count in the blood. So it's important to note that eating saturated fat, which is common in animal foods, very similarly mimics the biochemistry that we saw in familial hypercholesterolemia. So a fascinating story is there was a population that had a mutation in their PCSK9, where they essentially had hyperactive PCSK9 enzyme activity. And this enzyme degrades LDL. So essentially, this population with this genetic mutation had very little LDL receptor activity. So therefore, their LDL, their ApoB particles, were stuck in the blood. They had a very high residence time, they had a very high ApoB count, and they had very high coronary artery disease. However, then there was another population, a separate population, that had another mutation where essentially they had very low PCSK9 activity. So therefore, there was no degradation of the LDL receptor. So essentially, this patient population, their livers had a ton of LDL receptors. So therefore, whenever they secreted VLDL to form LDL, it immediately got sucked up back into the liver. So they had very low ApoB LDL counts, and they had very low coronary artery disease. So a lot of the evidence really points towards the importance of this ApoB count and this LDL receptor. And what's even more fascinating is we've developed medications that are essentially monoclonal antibodies that inhibit this enzyme. And when you inhibit this enzyme, you essentially inhibit the degradation of LDL receptor, so you have high LDL receptor activity, which can immediately suck up the ApoB containing particles, which should theoretically strongly reduce coronary artery disease. And there are even thoughts of creating vaccines to essentially to stimulate the immune system to create these monoclonal antibodies to essentially inhibit the PCSK9 activity, which personally I find very fascinating. So one last thing that I find very fascinating is that the NIH, the National Institutes of Health, they have a program called the Interventions Testing Program. And this is a very rigorous program that tests many different medications and interventions to see what actually extends lifespan. And I cannot emphasize enough that this intervention testing program is a very rigorous program, and almost every single drug or intervention that enters into the interventional testing program fails. They do not extend longevity. However, a few examples of the interventions that do extend longevity is rapamycin and metformin, basically an SGL2 inhibitor, and acarbose. And I find this very fascinating. The vast majority of the interventions fail, but a few of the interventions that actually do work seem to target type 2 diabetes. So targeting type 2 diabetes seems to extend longevity. And personally, I always found the idea of acarbose and an SGL2 inhibitor kind of interesting. You would slow the absorption of glucose, and the little bit of glucose you do absorb, you immediately pee out. So I always found that a fascinating concept. But why? Why do these type 2 diabetes medications seem to increase longevity? Well, of course, one thing is it reduces glucose. And we know glucose, the glucose molecule, goes through spontaneous, unregulated reactions where they glycate proteins. And when these proteins become glycated, they form these advanced glycated end products. So these proteins become dysfunctional. They have this bulky carbohydrate group attached to it. So these proteins become dysfunctional. So as you age, essentially your proteins stop functioning as properly. However, personally, I think what's even more important is that these type 2 diabetes medications, they reduce insulin. And I think that's even more important, and that's also related to why rapamycin extends longevity. Well, we know the three interventions that most consistently seem to extend longevity in virtually all animal models is the medication rapamycin. We also know that creating mutations in the IGF receptor extends longevity. And we also know calorie restriction, essentially eating fewer calories, also extends longevity. But why? What do these three things have in common? Well, we know they all inhibit mTOR, the mTOR protein complex that's in virtually every cell in our body. And if you're curious to learn more about mTOR, I have a video that goes into longevity and anti-aging science. But essentially, the three interventions that we see most consistently and reproducibly extending longevity in different animal models, they all inhibit mTOR. So therefore, you want to inhibit mTOR. When you inhibit mTOR, it upregulates pro-longevity genes. So again, we know when you inhibit mTOR, and it doesn't matter how, as long as you're inhibiting mTOR, you extend longevity and you extend lifespan. However, we know that hyperinsulinemia, and we know that insulin directly activates mTOR. It also essentially leads to increased amounts of effective free IGF, which also activates mTOR. And we also know branched chain amino acids can activate mTOR, but that's a topic for another video. But essentially, we know that high levels of insulin, which we see in type 2 diabetes, activates mTOR. And whenever you activate mTOR, that shortens lifespan. Because remember, it's when you inhibit mTOR do you get extended longevity. So therefore, when you activate mTOR, for example, in a type 2 diabetic hyperinsulinemia state, that shortens lifespan and that increases the onset of age-related diseases. So essentially, if you're sedentary and you're eating a lot of excess calories, you're going to have a lot of that intra-myocellular lipids in your skeletal muscle, and the skeletal muscle is going to be very insulin resistant. So that's going to lead to hyperinsulinemia and higher levels of insulin in the blood. And so that high levels of insulin in the blood, those insulin molecules are going to activate mTOR in the peripheral tissues in the body. And again, whenever you increase mTOR activity, you accelerate the rate of aging. However, if you work out, if you exercise, essentially your skeletal muscle becomes an energy burning machine. You'll go through mitochondrial biogenesis because you need so many mitochondria to burn energy because you're using up so much energy from exercising that you're going to be very insulin sensitive. And if you're very insulin sensitive, then you're going to have very little amounts of insulin. The pancreas only needs to produce very little amounts of insulin. So you're going to have very little insulin-activated mTOR activity in the peripheral tissues. And it's important to emphasize that high amounts of muscle is good for longevity for many reasons. Myokines are very complex, but we know play a role in longevity, and also the metabolic effects of having a lot of skeletal muscle. But personally, it's my belief that when you have skeletal muscle, we know that makes you very insulin sensitive, so we know that leads to very low levels of insulin. And then personally, I believe that low levels of insulin will lead to very little mTOR activity, which then should essentially mimic these other interventions that we know very reproducibly extend longevity and delay the onset of age-related medical conditions in many animal models. So again, if you're a type 2 diabetic, your skeletal muscle is going to be insulin resistant. That's going to lead to increased levels of insulin in the blood, which is going to activate mTOR in peripheral tissues. However, if you work out and have a lot of skeletal muscle, you're going to be insulin sensitive, which is going to reduce blood levels of insulin and is going to reduce overall activation of mTOR in your peripheral tissues. And I want to emphasize, metformin is not the end-all be-all when it comes to health. However, theoretically, metformin will help a type 2 diabetic become more insulin sensitive, which should reduce blood levels of insulin and therefore collateral activation of mTOR. So theoretically, metformin may help a type 2 diabetic have more average levels of insulin sensitivity and insulin levels. However, it's not the same as if you work out. If you work out, you're going to be more insulin sensitive, have reduced levels of insulin, plus all the benefits that skeletal muscle brings when it comes to general health. One last thing to consider is that insulin is a growth factor. So, in order to develop a full-blown cancer, we know a healthy cell must accumulate several mutations before it develops a full-blown malignancy cancer. However, that should not happen. For example, when you have a precancerous cell, this cell is balancing pro-apoptosis versus anti-apoptosis. And this cell, with these DNA mutations, it should activate apoptosis. We should get rid of this cell so it does not develop more mutations to form a full-blown cancer. However, when we have this precancerous cell, and you're constantly activating mTOR, you're activating growth. You're enabling this precancerous cell to continue to grow and develop more mutations to develop a full-blown cancer. And again, insulin is one of the key activators of mTOR, which allows this precancerous cell to tip the scales towards anti-apoptosis and pro-growth to allow the cell to continue to divide and accumulate mutations. However, on the flip side, if you stabilize p53, you provide a robust cellular surveillance system to readily activate apoptosis once a cell is exposed to enough DNA mutations. For example, right now, your pancreas may have precancerous lesions. However, this precancerous cell is debating whether it should promote growth and allow the cell to keep on growing, or if it should just activate apoptosis to get rid of it so it doesn't develop the chances to develop more mutations. So therefore, theoretically, if you promote robust cellular surveillance systems by directly stabilizing and upregulating p53, you tip the scales towards pro-apoptosis to readily activate apoptosis once a cell accumulates enough DNA mutations and cellular stress. And if you think about it from an evolutionary perspective, it's very energetically costly to activate apoptosis. If we activate apoptosis and get rid of the cell, it costs a lot of cellular energy to develop new cells to replace it. So in the scale of evolution, it makes sense why we never promoted this pro-apoptosis state, because it was so energetically costly. However, now in our current day and age, where energy is not as much of an issue, it makes sense to stabilize p53 to activate pro-apoptosis systems in order to prevent the onset of cancer. And we do have other interesting technology that essentially analyzes the DNA in the blood. For example, if you have a pancreatic cancer right now, some of those cells are going to activate apoptosis, and they're going to release the cancerous DNA into the blood. And you can take a blood sample and analyze all the DNA in the blood to see the patterns in order to predict whether you have a very early cancer. So this technology exists and theoretically could help prevent and catch early cancers. So the last thing I want to mention is please eat biology. Eat food that's made out of real cells, because we know real cells contain all the nutrients, the vitamins, the minerals that you need for cells to function properly. All the minerals and cofactors you need for proper health. And not just that, we know when you eat real biology made out of real cells, you also get fiber. And fiber essentially helps increase satiation and it makes you feel more full. Or you can eat something that's made out of a chemistry lab. And something that's made out of chemistry lab is basically just salt, fats, and carbs. And not just that, there are billions of dollars that are spent to create food that hijack the satiation centers, especially in genetically susceptible people, which essentially hijack the satiation centers, which make you want to eat more food. So, just simply, I try to eat more biology at least a few times a day, eating real food made out of real cells with all the cofactors and nutrients you need for proper health, and also fiber.